Hello everyone. I would like to introduce our presenter today, Alcine. She is pursuing a master on the bio-fertilization with specialization in crop physiology, production, and horticulture. She graduated from Earth University in Costa Rica with a bachelor degree in agriculture science and natural resource management, promotion 2019 to 2022. And her thesis was based on the correlation of bricks reading with shelf life of berries by photostimulation under controlled and protected environments. Her presentation today will be about tryptophan and lysine content of two quality protein maize varieties in two Luvisol with bio-fertilization in Yucatan, Mexico.
Give it some energy. Come on.
Thank you so much, Lorena, for this warm introduction. And yeah, I'm going to present an article today, and they did a research on lysine, yield, tryptophan content of two quality protein maize in two Luvisols with bio-fertilization in Yucatan, Mexico. So you might ask the question, why this article? So I'm just in first year in my research thesis, and my project will be exploring the agronomic and economic potential of quality protein maize, sweet corn, popcorn, and dead corn varieties in intercropped with legume cover crops here in Nebraska. And we aimed to evaluate the yield component and compare them with normal versus quality protein maize, and also with our no cover crops under organic versus conventional system. So when compared this research done in Mexico with my project, we are both going to work with quality protein maize. And in this article, we can see the importance of quality protein maize, QPM. That is really an important asset right now to help combat food security in the world. So let's start with a quick introduction. So we know that native corn varieties in Yucatan, Mexico are really important because they are staple food, and they have been planted annually in around 390,000 hectares. And even if they are really important, this native corn still has low yield potential and also poor protein, and this affects because as a staple food, the population can have a higher risk of malnutrition. And add to this, the soils in Yucatan, they are really heterogeneous due to intensive cultivation, the high amount of fertilizer used. So this gives an importance or a boost to quality protein maize, because why? The quality protein maize, as seen in this graph here, they have done a comparison between the normal maize and the quality protein maize varieties for nutritional parameters, and then they have found that lysine and tryptophan, they are two amino acids important for protein content in maize, and they found that they could have twice the amount found in normal maize for these two amino acids. So these quality protein maize varieties are around the world right now that can help to improve nutritional status, also to help benefit for food security and sustainable agriculture. Another background about QPM varieties, they have been developed in the 60s, and the principal objective was to improve the efficiency in lysine and tryptophan. The two amino acids are more important that can be found in maize. And in 2010, INAFAB, that is an organization in Mexico, they found and launched into the market two quality protein maize called Chichen Itza and Sagbe. And they have done researches around the world to evaluate protein quality in different corn materials and also in different conditions. However, they have done no research in these two quality protein maize. This is why the research for this article, they wanted to evaluate the yield performance and also found lysine and tryptophan content in Chichen Itza and Sagbe in different intensive use Luvisol, and also to assess the effect of applying chemical fertilizer alone or combined with biofertilizer on yield and protein quality. For the materials and methods of this research, they conducted the research in the south part of the state. This point here is less or more where the experimental station was ubicated, and they did it in spring summer 2017 with two different soil type, as I mentioned before, and they did moderate fertility. The altitude there for this station was around 50 meters above sea level, and the precipitation and temperature annually average around 900 millimeters and 25 degrees Celsius respectively. And the two varieties mentioned before, Sagbe and Chichen Itza, were the one evaluated or studied, and they had low and high intensive agricultural use. And I'm going to explain that in this slide. So in this research, they're going to mention lot 1 and lot 2 for the result content and for better understanding. And what is a lot 1? Lot 1 is the low agricultural intensive use. And why? Because the corn has been planted there sporadically for every 4 to 5 years with no longer fallow periods. And compared to the lot 2, where they have the high agricultural intensive use, and the corn has been grown every year here for more than 20 years without any fallow periods, and they also applied fertilizer and irrigation in this lot. So that's why the contrast for soil here is going to be really different for the two lots, and we're going to see that affect or influence the results that we're going to have. And also we're going to see that because the salinity and phosphorus in these two soils are really different. The next one, we're going to talk about the experimental design. So the treatment that they use here, there were chemical fertilization, bio-fertilization, and the control that I'm going to explain later in another slide. And they had the two different Luvisol conditions, the lot 1 and lot 2, as explained before. And they did it in a randomized complete block design with 3 replication. And the plots dimension were 5 meters by 4, and with 4 walls distanced with 5 meters for a total population of 50,000 plants per acre. The treatment. Here we can see the 6 treatments that they used. So they had 2 control basically because they separated the varieties. One control for the Chichen Itza and the other one for Sagbe. And those other treatments for chemical fertilization, they added the 60-80-00 for like the varieties separately as well. And also in the last part, that is the T5 and T6, they had the chemical fertilization, but they added a supplement as a bio-fertilization. And in here they use the mycorrhizae and azospirillum as a bio-fertilization. And the parameter measured in this research, they focused on yield of grain. They measured a ton per hectare, the tryptophan content in percentage in proteins, lysine content in percentage in proteins, ashes in percentage as well. For the statistical analysis, they did an analysis of variance, and they put a 2K test for confidence interval at the 95%, and for the confidence of information, they found it by calculating the confidence of variation. Let's start with the result. So here we're going to start with the yield performance, and in this part we could see in this table that the higher yields performance were in the bio-fertilization, as we can see here, and the Chichen Itza was the variety with higher productivity. And as we can see, the take-chem bio, it means that for the chemical fertilization plus the bio-fertilization. And we can see for lot 1, Chichen Itza, and for the lot 2, the Chichen Itza had higher performance for only the chemical fertilization, and for lot 1 in Sac Bay, the chemical bio-fertilization still had higher performance for the variety Sac Bay, and also for the chemical application and lot 2 as for the Chichen Itza. And for the protein content here, we can see that the Chichen Itza for lot 1 in the biochemical application plus the fertilization, the chemical fertilization had the higher value for crude protein. And also we can see the Sac Bay didn't really act well in all the treatments and all the areas, and we can even see the control had a tendency to be higher in crude protein compared to the Sac Bay in lot 1. And in grain ashes, we saw that the Sac Bay in this one presented the higher value for both lot 1 and lot 2, 2.01 and 2.22, and the greater assimilation for Sac Bay is still in both lot 1 and lot 2, and the highest value was found when applying the chemical fertilization in both lot 1 and lot 2, and lowest value was found for Chichen Itza in the lot 2. The protein content, we're going to start with lysine. There were no statistical differences between Chichen Itza or the other variety Sac Bay. However, we can see in these graphs, in this table, sorry, that Sac Bay has a tendency to be higher than the Chichen Itza in lysine content protein. For tryptophan, the Sac Bay also presented a higher value for the chemical application plus the biofertilization for in all the lot 1 and lot 2 regardless of the treatment, and the Sac Bay had a 42% more tryptophan than the Chichen Itza. Okay, so now let's talk about the discussion and a conclusion. We could see that in this research, the QPM varieties and the yield component for these varieties and the nutritional quality were enhanced by biofertilization, as we could see in the graphs and tables that the biofertilization had higher value for hashes, crude protein compared to the other treatments. And also we saw that these researches with Luvisol's properties had a higher impact in maize performance, because when we saw the tendency for yield performance, both varieties were doing well in this concept. And we could see that the quality protein maize could help with sustainable agriculture, because when applying biofertilization in the lands, it helped to reduce fertilization and also to eliminate disease and insects and all of these, and also can help to improve food security. And when you think about the regions where maize is the staple food most used, this can help to improve the protein deficiency in the diet in this population. And also for this research, the Sac Bay was shown to have a better protein quality than Chichen Itza. However, the Chichen Itza presented the highest value for yield component in this research. That would be all. Thank you.
Okay, thank you. Any questions?
I have a simple one. I guess you were talking about the measurements you were using. Like you mentioned ashes percentage. Is that, I mean, I'm just not familiar. Are they like burning it, or what does that mean?
Also, in this research, what they did, they burned the biomass
Okay.
to find the quantity of ashes they
Okay.
have for this one.
So they're just trying to find the biomass.
Yes,
site.
the
Right,
biomass.
okay. Gotcha. It's like
Yeah.
in burning. Okay.
Yeah.
So any of the phenotypes plants were affected by having the high lysine and high tryptophan in them. It's like a negative effect. When you increase the high lysine and tryptophan, the plants still look normal.
So in this research, they did an inoculation for the seeds before planting it to help with too much pollination, because they said that maybe the varieties might have like I don't know, it's a superpower to attract a pollination or not attract it when like in the same line with other conventional maize, I say it like that. But however, I was talking like with some doctors that I met last time I went in a conference. I was presenting a background about my research, and they were mentioned that actually lysine and tryptophan are really important for human consumption, and also if it can be used in animal feeding as well, that would be great, because that they are two really important amino acids.
I understand, but do the plants suffer any negative aspect of having high lysine and tryptophans, like lodging? Do the plant lodge more when they are high in lysine and tryptophan? Because in normal corn doesn't have high lysine and tryptophan. So when you start manipulating the genetic components of it, is there any negative aspect of it? Do you know?
I really don't know exactly if we could have a negative impact, but the earlier researches that they did when adding more lysine and tryptophan, they found like I don't know if the
So even also the kernels,
skin?
sometimes the
Yeah.
kernels get soft.
Yeah, they said
Because of
something
the-
like that. But they did another implementation, and they found like they improved the how do you say this one again?
Kernels?
Kernels, yeah, of the maize, and it has been improved in not being so soft compared to the first varieties that they developed.
How did they do that? Do you know?
I did.
Well, it's like-
I don't know
okay.
anything about genetics.
It is, yeah. It's
I
like,
don't know about that, unfortunately.
Okay.
But I will certainly read more about it, and maybe next time I have an
Any other
answer.
questions? All right. You know, there is a movie after your name. I saw it in the library. Yeah.
I didn't.
Maybe I should bring it next week. Yeah, exactly your name.
Well, I'll have to be honest.
Okay.
Yeah.
Maybe after your tribe.
Yeah, so I enjoyed the presentation, and I don't know, maybe a bit biased because one of my colleagues in my lab is actually working on QP, vegetation components. Yeah. So the fact that the plants are having the high lysine content in the tryptophan, it doesn't affect the plants negatively. Yeah, from his research so far. Yeah. It doesn't really affect. I think it affects the conversion of, I think, storage, the kernels. Yeah, I think it affects the kernels, but not in a negative way. So he's actually using the QPM for
Okay. So, yeah, how about the presentation?
Yeah, for the presentation, yeah, I enjoyed the use of colors and then the high quality visuals. I think the pictures were like very high quality. Yeah, then also the flow, the general flow, I really enjoyed it. My only comment would be the starting point. It felt like you were a bit, like you wanted to be the time, so that was how I felt.
Okay, no other comments, suggestions? Thank you. All right.